So now we're in the second part of our meeting, our seminar. So all the David Kasahara and John Godlez have been a part of our program, receiving students at their labs. I'm very happy to introduce Quan Lu. Quan Lu is a PI there. It's the first time that he's receiving a student there. We hope you guys are very good at his lab for him to be able to receive more students and be an example to the other PIs as well. So Quan Lu has a bachelor and master's in biology at Fundang University in Shanghai, China. He had his PhD at Iowa State University, and he had his postdoc at Stanford University. Currently, he's an associate professor of environmental genetics and pathophysiology at the Department of Environmental Health at Harvard School of Public Health, and he's a new professor in our program. So thank you and welcome to University of São Paulo.
Thank you, Lucas. So good morning, everybody. So my lab actually is relatively young, I guess. So I moved from Stanford, California, to my current position in 2007, I guess. So it has been actually 7 years. It's not that young anymore. But so the point is that I was trained actually as a molecular biologist, a cell biologist, and functional genetics. So my lab actually used a lot of this tool, genetics and genomics tool to study a very basic cell biology question. So because we are at the School of Public Health, so there are lots of interesting figuring out the human diseases that are highly relevant to public health. So being in the Department of Environmental Health, so if you think about this one, so all the major human diseases that exist today, cancer, type 2 diabetes, asthma, they are all complex diseases. They are not simple, like one gene, in one disease, like cystic fibrosis, which is determined by mutation in the CFTR gene. So all the major diseases, actually, they are complex diseases. When I say complex disease, not only because at the molecular level, they are not just one gene, one disease. Actually, cancer, type 2 diabetes are, many, up to hundreds of genes actually contribute to the outcome, of the disease. And then, in addition to this, complexity at the DNA level, there are also all these complex human diseases. There's a major influence from the environment. I put this picture of air pollution here, but in addition to air pollution, you can think about anything like the contamination in the water, the nutrient status, what you eat. For example, in type 2 diabetes, if you eat fast food, a lot of sugars that can contribute to the disease. So the combination of the genetics and the environment actually lead to the development of all these complex human diseases. So my lab is very interesting figuring out the interaction between the genes and the environment, how that interaction leads to the major human disease. So how do you do that? I mean, there are many approaches you can tackle this very complex problem. So my lab is taking I call it a very simple-minded approach, really simple-minded. So what we do is that we start with because of the disease, so we can identify the hallmarks of the disease. For example, in asthma, a lot of time is inability of the airway to open. So that's a feature, biological feature you can identify. Then we design a cell-based assay to model that phenotype. Sometimes it's oversimplified, but it's good to have some place you can begin with. Begin with something very simple. And then what we do is that once we have this robust cell-based model, and then we can do functional genetics or genomics in this cellular model, identify genes or genetic pathways that contribute to this phenotype. So once we have this is a very basic cell biology and molecular genetics. Once we do this one, we obviously will end up with maybe dozens or hundreds of genes that are important for this cellular phenotype. And then, so once we have this list of genes, what we do is that we do mechanistic studies, molecular cell biology, biochemistry, try to figure out why these genes are important for that particular cellular phenotype that's relevant to the disease. Okay, and so this will take a bulk of our in time. Most of us our time will spend to figure out the molecular mechanisms of these genes in the cell biology, in biochemistry, biochemistry aspect of this. But so our lab actually focuses here and here. So and then once we have these genes and the biochemistry in the pathway figure out, then we ask whether these genes or the genetic pathway are relevant for the human disease. We look for genetic variants in the genes we identify to see whether there are particular variants may predict somebody may become more susceptible or less susceptible or resistant to certain phenotype. And also we can link to the animal models. If we figure out a gene or pathway that's particularly important for for disease, we can do transgenic or knockout animal models to further study of that gene in that in that phenotype. And as you can see, these links are bidirectional. So, for example, in the human population, we can feed in the genes pathway to the human studies. But also, once we start do studies in humans, then we can feed the information back to the basic in biology. For example, if we identify one particular variance that in certain population makes this population more susceptible to certain disease, then we go back to the molecular biology to ask why this in particular variance. For example, does this variance impact the expression of the gene or function of that gene? So that's the approach. This is the general approach, and I call it simple-minded because this is simple-minded because we believe that there's a fundamental underlying genetics for all these complex human diseases. So today I'm going to tell you two short stories that to illustrate this approach, this simple-minded approach. One is about this arsenic exposure, and how that exposure to arsenic, particularly in drinking water, lead to the type 2 diabetes. What's the mechanism of that? And in the second story, I tell you, related to asthma therapy about this receptor called beta-2 adrenergic receptor, or beta-2 AR. Or B2AR, as we call it simply. So for the first story, so arsenic, some of you may know it, and actually it is a poison, but here we are not talking about arsenic as a poison, but rather arsenic as a toxic matter in contaminants in the environment. It's very ubiquitous in the environment because it's happening in the mining, and also very important for public health is that many people estimate actually over 150 million people worldwide are exposed to high arsenic in the drinking water. So a lot of the people, actually, particularly in Asia, also in the U.S., I believe also in Brazil, but I'm not in a particular very clear on this point. But exposure to arsenic, particularly the chronic exposure to arsenic, has been associated with a lot of diseases, skin lesion, cancers, all kinds of cancers, and also type 2 diabetes. So that's our focus. At the cellular level, what arsenic does is to damage the protein. So it reacts to the side group of protein, therefore causes the protein misfolding. That once you have protein misfolding, the cell have to respond. Then that response called endoplasmic reticulum stress. ER is the site where the protein are being synthesized. So if there's a misfolded protein, the cells activate this ER stress response, and then tell itself to either to correct this misfolding problem, or if that cannot be corrected, and then the cells become apoptotic to commit suicide. So the ER stress is very complicated. There's lots of studies from the East. From humans that figure out all these biochemical checkpoints of ER stress pathway. I'm not going to go through this process, but I'm just to point out that a lot of these pathways are leading to this activation of CHOP, which is a transcription factor, that if this CHOP is activated, the cells commit to the suicide called apoptosis. So what we try to figure out is how arsenic actually exposure actually activate this process. So we do, as I said, we do functional genetic screen. So the idea is very simple here. You begin with the target cells, which is a Y type. Okay? So and then we do genome-wide inactivation. This is called RNA interference. So this is the method we use, RNA interference. And so what it does is that it will inactivate each gene in the human genome one at a time. So basically, at the end, you create a library of cells in which these cells have a gene X being activated, these have a gene Y being activated. And then you ask from this population of cells, and what's this mutant cells, for example, this changed phenotype, for example, alter response to toxicants. So normally, if these cells are all sensitive to certain toxicants, here you isolate the cells that are resistant, meaning that they can withstand the insult from these, from the chemical toxicity. Once we identify these mutants, then we can go back to see which genes are inactivated in these cells that lead to that phenotype. So that way we can identify the genetics behind the toxicity again. So that's a very simple idea. So I want to introduce to the concept of RNA interference. So some of you may know if you are familiar with the very basic genetics. So this is a method whose discovery actually lead to the award of Nobel Prize about 5 or 10 years ago. I think probably 10 years ago. So the idea is that there are double-strand small RNA molecules that lead to the suppression of the gene expression. Okay? So I'm not going to go through, but if you're interested, you just type in RNA interference, and you can Google it. There's lots of information about this, and you can figure out how this RNA interference really works in the cells. So in fact, this is an endogenous mechanism they call microRNAs that's used by the cells, by all the your cells in the body, the worm cells, mouse cells that used to suppress their gene expression. Okay? So what we do is that we generate this construct these large libraries of RNA molecular known as short hairpin RNA molecules, and then we do the selection as I mentioned a couple of slides ago, and then identify the mutant cells, and then do DNA sequencing to find out what the inactivation that happened in that particular cells that lead to that phenotype. How we do the arsenic ER stress RNA screen. I'm not going to go through this, but essentially, as I mentioned, you have to have a robust cellular model that you can assay for that phenotype. That's very important. Once you establish this cell model, and then you can transfect infected cells with the library, and then establish a genome-wide RNA knockout library, and then you separate the cells based on certain phenotype. In here, we use a flow cytometry based method, as you can see from the next two slides, and then we recover the cells and their DNA, and then do DNA sequencing, identify the genes, and then we do validation. So here is the cellular assay we use. So the idea is that we fuse this M cherry, which is a fluorescent protein, red, okay, to a promoter, which is CHOP. If you still remember, the CHOP is the gene that in the ER stress pathway, that at the end point of ER stress pathway, activated by the ER stress. Okay? So the idea is that this is a construct we stably put into the cells. The idea is that without arsenic treatment, there's no activation of CHOP. Therefore, no expression of M cherry. The cells are dark. You don't see anything under a microscope. And if you activate the cells with arsenic, so it basically treats the cells with a toxic, in this case, arsenic, Then this will activate the CHOP promoter. Therefore, you have emission expressed. So look under the microscope, you can see red, red fluorescence. So here that we establish this clone. So basically, this flow cytometry. So on here is the number of cells. Here is this amount of emission expression. So normally you don't see so this autofluorescence, some background fluorescence. But if you activate the cells with arsenic, as you can see, the fluorescence gets a dose-dependent increase, okay? Up to 30 micromolar, you can see. And there, at the end, we select 15 micromolar as our screening condition because it leads to a very low cell toxicity. So here's a screen we do. You have a bunch of cells, this library, activating about 10,000 human genes. And then treat it, and this will normally activate the expression of the fluorescent. And in this population, we look for cells that do not get activated by this. So the idea is that these potentially contain a gene inactivating events that lead to the disruption of the activation. So you do this, and you can sequence the clones identified from the flow cytometry sorting analysis. So without going into the details, here we identify at the time we sequenced about 250 shRNA in clones. 60% of them corresponding to known genes. Known gene meaning they have name, they have certain description of the genes. And 40% of them, some of them, for example, they are non-coding. As some of you know, that a lot of genome regions, they produce RNA molecules, but they do not code for proteins, but still they are functional. So they're called non-coding RNAs. Then there are some mixed clones. Then there's ESTs, meaning still that they have some express sequence tag, but they don't know what the genes are for. So initially we focus on the known genes because at least there's some biochemistry can follow. So this list of known genes, and I'm not going to go into detail, but we have from this list of genes we can do pathway analysis. So if you have hundreds of genes, it's hard to see why, what's the process that's being impacted. If you want to see a common process, you can put these genes into a pathway analysis. So the bioinformatics analysis will tell you what kind of pathways these genes are involved in. So therefore give you a general idea about what in process are being impacted in these cells. So the number one is the intracellular stress signaling, which is expected because this is an ER stress screen. Okay, so you expect a lot of genes from the stress signaling, including IRE1, which is the canonical gene that's normally inactivated by ER stress, and also a bunch of other genes, oxidative reduction, blah blah blah. So the first study we focused on this gene because it's related to stress signaling and it's normal. But turns out this is amino acid sensing or amino acid transporter. So we did the study that if you knock down these genes and then here, so this is the shRNA, this siRNA, which is a short interfering RNA. So this is a reagent that you use to initiate the RNAi process. So if you treat the cells with RNAi, then you can see their response to arsenic is reduced here, and then this is a knockdown of the gene. And also, in addition to CHOP, and another ER stress marker, XBP1, also gets reduced if you reduce the SNAT2 expression. So I'm not going to go through this, but this is more biochemistry. Figure out how what genes or proteins are activated, inactivated in this process. Okay. For summary, the first story is that remember the idea is that you have to have a robust cellular model. So here we introduce the arsenic-induced ER stress, use the CHOP M-cherry fusion protein as a model, and we perform a genome-wide RNA screen and identify both known and novel regulators of this stress response. And we characterize SNAT2, and obviously the lab is still interested in other RNA-seq hits that and figure out what the biochemistry and genetic pathways these genes are impacting. So in the second story is about I think David, give a very nice introduction to asthma. So this is has something to do with asthma pharmacogenetics, or asthma therapy. And so the hallmarks of asthma, there's probably 3 of them. Airway inflammation, particularly of the airway epithelium. And then David mentioned about airway remodeling, mostly involved in the smooth muscle cells. And for example, smooth muscle cell gets hyper, proliferative, and therefore lead to the increase of the smooth muscle mass in the airway. And then also induce the changes in the contractile and secretory pathway, in the phenotypes in the smooth muscle cells. Also, the airway become hyper responsive to allergen treatment, in a lot of things. That's why this is a hallmark of asthma. And this is just show you the normal airway versus asthmatic airway. So, in the mainstay of asthma therapy is this beta agonist, for example, like albuterol in the inhalers. So that they used to, the mechanism of this, how beta agonists such as albuterol works is that they activate the receptor called beta-2 adrenergic receptor in the airway, particularly in the airway smooth muscle cells. And that leads to the biochemical pathway that relaxes the airway smooth muscle cells. Therefore, you have bronchial dilation, and that allows the individual to breathe. So this is a receptor that the albuterol and other beta agonists is activating. So this is a plasma membrane, and this the beta-2 receptor is a 7 transmembrane protein. 7 transmembrane meaning they have spanned in the membrane 7 times. Okay, so 1, 2, 3, I guess 7 times. So, and actually the discovery of a beta-2 adrenergic receptor, and along with this group of protein called G protein coupled receptor superfamily, actually, this work was awarded the Nobel Prize in chemistry about 3 years ago. So this is still a very active field of research. So the beta agonist, once they bind to the beta-2 receptor at the plasma membrane, activate the receptor. That's lead to the recruitment of this G protein. This beta-2 receptor, particularly to the GS, which is stands for G protein, stimulatory protein, that lead to the increase of cyclic AMP, and that can activate the protein kinase A, and then induce the relaxation of the airway smooth muscle cells. That's how all the beta agonists worked. So, previous work has shown that the agonists actually can induce this beta, not only activated the receptor, which lead to the smooth muscle relaxation, but also that lead to a process called receptor downregulation. So actually, this is a good process normally, because you don't want to have the cells to have this persistent signaling, which would be bad. You have, airway always relaxed. That's bad. So normally, this process, once it's activated, it's almost immediately, being downregulated. Therefore, so if you look at this one, so this is the cells artificially express this beta-2 receptor with a tag at the surface. You can see the receptor very nicely expressed at the surface. If you give the cells isoproterenol, which is a beta agonist, this leads to the very dramatic downregulation very quickly, within in 15 minutes. So over the years, people have figured out a lot of genes and pathways are involved in here. So what we want to do is we ask whether we can identify new regulators of this process that may be able to enhance the beta-2 receptor in the therapy for asthma. So again, back to my simple-minded approach is that we need a very robust cellular assay. So what we designed is that we express the tagged receptors. Here we use flag tag. So this is a short amino acid you can attach to a protein. Therefore, later on you can follow, where the protein goes in the cells. So flag tagged as this green transmembrane protein at the cell surface. And the idea is that if you give the cells isoproterenol, this will lead to the normal downregulation, as I show you in the previous slides. So now we come in with a genome-wide RNAi library, so that will inactivate the genes at the genome-wide level. Okay, so we have thousands of genes being inactivated in a collection of library. And then we do the same process. So normally, if the gene has nothing to do with beta-2 receptor downregulation, then it will behave normally. Okay? The receptor will be degraded. If a gene is essential or extremely important for that downregulating process, when you block it by the RNAi, and then the receptor cannot be degraded, okay? They lead to the increase of this tagged receptor in the cells. So by doing the very simple flow cytometry sorting, then you can identify this population of cells because they have higher amount of this receptor. And then once you have these mutant clones, you can identify what is the RNAi reagent that lead to the phenotype by simple DNA sequencing. I'm not going to go through this process, but it's just show we have stable cells that express this receptor, introduced with a genome-wide RNA library, induced with isoproterenol, stained to quantify the amount of receptor at the surface, and then essentially look for cells that still retain the receptor in the presence of beta agonist. Okay, these are likely in mutant cells, and so we can either go to grow single clones, or we can just get a mixture of FISI positive cells, do DNA sequencing, verified by retransfection. Makes long story short, that's a list of genes we identified. This is not the very exhaustive list, but we have published papers on the first one, arrestin domain containing protein. At the time when we identified that it's a novel protein, nobody knows what's the function. So it's the first time that we assign a very important process, beta-2 receptor downregulating, to that gene's function. And we also identified this gene called farnesyl diphosphate in the synthesis, which, as the next slide will show, that the reason why we are interested in here. So we have published work that show that FDPS is required for the beta-2 receptor downregulation. It means that if you block it, either through the RNAi, means knocking down the gene expression, or through this very specific chemical inhibitor called alendronate, which previously several pharmaceutical companies have identified this alendronate as a very, very specific inhibitor of FDPS. So if you block this one, we know that will block the beta-2 receptor downregulation through the internalization process. But more interestingly, because this alendronate, in fact, is a drug that's used to treat a human disease called osteoporosis, particularly in elderly women. So the idea is that if this is required for this beta-2 receptor downregulation, and you can inhibit this enzyme through this chemical inhibitor, what if is it possible to use alendronate for the asthma therapy? So the idea is that if you give a patient alendronate with beta agonist, normally beta agonist will induce the downregulation, particularly the long-term use of beta agonist will lead to the inefficiency, the reduced efficacy of beta agonist in asthma patient. Here, if you give a patient also the alendronate, if we can stabilize the beta-2 receptor, increase the amount of beta-2 receptor, therefore, in these patients who receive alendronate plus beta agonist, you will have a better response to beta agonist in terms of their bronchodilation in response. So that's the thing that actually Elliot Israel, my collaborator at the Harvard Medical School, who is also part of the NIH asthma network, is actually this pilot clinical trial has been approved by NIH. So we're actually in the process of recruiting patients. This is a small pilot trial. We will have about 80 patients. Then at some point we will do randomization, and then one group will do. So this is a placebo arm, and then this is a real arm. So basically, the patient will receive inhaled, corticosteroid therapy plus the long-acting beta agonist. In the placebo, they only receive these two combinations. But in the active group, they also receive alendronate. So at the end, we'll compare their lung function as an indicator of their response to beta agonist. So hopefully probably by middle of next year, we'll know the result. But this illustrates that in a point, very basic cell biology or genetics research can potentially lead to the improvement of therapeutics for patients. So really, that I think this, to me, I think that the basic research is a driving force of a lot of these new therapies. Because without basic research, you can only rely on the old therapies. So I think this is a point that I want to make to you, because particularly many or all of you are going to be physicians to practice medicine, but you have to keep in mind that basic research is extremely important. So the other thing that, as I mentioned, that we want to get to the human, in the previous slide shows that there's a potential to improve therapy. But the other point is that we can ask whether in the genes we identify in the RNAi screening hits, whether there are genetic variants in the genes, in these genes that may make cells, make humans more susceptible or less susceptible to the therapy. In this case, we use bronchodilator response or BDR as an indicator for the asthmatics. And indeed, we were able to identify several genetic variants in the genes we identify that are associated with BDR response in asthmatics. So we are actively following up on several of these SNPs with our collaborator Kellington Teixeira and Scott Weiss at the Channing Lab. So to summarize, that this is we developed. I did not show you actually the RNAi library. Actually, we designed in the lab. It's called ESD-based library. We use this library we designed and did the RNAi screening, identify a number of regulators of beta-2 adrenergic receptor, and we characterize FDPS, and we show that there's a potential to use FDPS inhibitor as additional therapy to improve beta agonist response in asthmatics. I did not have time to go through one of the non-coding RNAs that we identified. We named it fresh air because it potentially can improve the bronchodilator in the airway smooth muscle cells. And there are genetic variants in the RNAi hits we identified associated with response in the patients. So acknowledge people in my lab and our collaborators and the funding sources. So thank you.
So we have time for questions. So do anybody have a question? I have a question for you.
Yeah.
It's about the kind of work that the students more about the program. You are not a new PI. In fact, you're a new PI to join your program.
Right.
Yeah. So what the type of work people are doing in your lab, you think, related to asthma and related to this alpha project or related to other projects? What do you envision as?
Right. So people in the lab, I think that you will learn a lot of this very basic cell biology, like cell culturing, immune staining, imaging, like fluorescent imaging, cell sorting. So this is a very basic cell biology. I think it's fundamental to a lot of these sciences. And then you will learn how to do, perform genome-wide screen. Although probably I'm not going to give you a project that asks you to start from the very beginning, but you will get exposed to a lot of these screening projects in the lab. So the, genetics, functional genetics, particular RNAi approaches, short hairpin RNA moleculars, as well as this new, I did not have time to tell you some of this new work we are doing, use this so-called, the CRISPR, very precise genome editing tools have been developed in the past year and has been a huge, in addition to the tools that functional genetics people use, So that also you will be able to learn that new technique. We do a lot of protein biochemistry, meaning in the Western blotting, and we do very basic molecular biology like DNA cloning, transformation, sequencing, and we also have also my lab is not active in doing this bioinformatics, but a lot of our work involve a lot of use of biomedical research tools. How to analyze big data, because a lot of these sequencing data actually. So I show you some of the just simple single DNA sequencing, but now because of this RNA-seq or deep sequencing technology, we use a lot of deep sequencing. So that involves your basic knowledge about how to use computer programs to really analyze this data. So in the lab, we will be exposed to that kind of things. So I think it's depending on the project. There, you will, I think that yeah. So that's pretty much. I think the year that what. The reason I did not participate in John's program, although I know personally, have know that being very impressed by a lot of your work at HSPH from Brazilian students. Because previous we don't have a lot of senior people in the lab to mentor new students, but now I think it's a good time for our lab has been stabilized, with stable funding, with a lot of several senior people listed here, that some of them can serve as a mentor that in the lab for you directly. Because I mean, if I have time, I would like to work with you, but a lot of time I don't have time to personally tell you how to do stuff in the lab. But these people will be perfect mentors. Some of them are research scientists. There are senior grad students in the lab. They have been in the lab for 4 or 5 years, So, yeah.
Do you have any other questions? Any other comments from any student? Any questions about the program or about questions? No? Yeah. Anything else? No? Okay.
Great.
So we'd like first to thank you, Quinn, and to thank you, David and John. So in the name of the alumni, I would like to thank Paulo Saldiva for bringing up everything and to receiving the professors here, Luis Burns, who are always helping us, and this year especially Rodolfo, who is leading the alumni, organizing everything. So this is something we expect you in the future to have. So we expect you in the future to be having Rodolfo's role of organizing, receiving the professors, having the course, doing research in Boston. And so thank you, Rodolfo, for helping Andressa as well. She was here, she helped Rodolfo organizing, and we expect you to be doing this in the future. So have good interviews this afternoon, and tomorrow we hope to have a good group for 2015. And for the students who are not selected, please consider applying again next year. We want bright students and students with initiative to be part of this program. So thank you all. Yeah.